Hi, I'm Dr. M. Kamakshay, open source software evangelist and also a full stack developer. I develop full stack applications to help my students learn data science and analytics, and also to help a couple of my fellow academics, and also a couple of software development firms located in India and abroad. The purpose of this video is to make a small demonstration on a very small application which I developed just recently. I call it as GAtRS, which stands for Genomic Analysis through R-Shiny. My internet connection is very slow. It's this one, GAtRS, so Genomic Analysis through R-Shiny, and the file. This is a file which is very important, and which we before going to the nitty-gritty of this application. Why I started developing this application? Because I just came across. I know today we are in a very big world crisis, health crisis, that is the pandemic, COVID-19, novel corona, virus pandemic all over the world. Right now I am living in a city which is under lockdown. I just started studying the gene sequence of, I wanted to study the gene of COVID-19, which is novel corona virus, and sooner I felt that it is better to develop an application for other individuals who can also perform some kind of this application is not just for coronavirus. It can be used for any, as long as you have a gene sequence in your hands, you can just make use of this application to perform analysis on that gene sequence. So coming back to the point why I started developing this application, because I just came across a statement somewhere in one blog that COVID-19 genome sequence is 79% similar to SARS-CoV and 51.8% similar to MERS-CoV. This seemed ridiculous to me, because the genetic distance between any two viruses must be very minimum, which means, like, suppose if we take genome sequence from two same species, two different creatures of the same species, then it should match 99.99%. For instance, if you take gene sequence of a human being and you match with some other your fellow neighbor gene sequence, then the similarity should be 99.99%, because both of you are Homo sapiens, and your gene, the source of your gene, our gene, is same. So in the same fashion, if you take two plants and the gene must match, like it doesn't matter whether it is apple or banana or whatever it is, the fruit, the category of the plant, if you take the gene sequences of two different fruit plants and do analysis, then the gene sequence must match 99.99%, because both of them share the gene from the same source. And now if you take the gene sequence of any two bacteria, for that matter, the human gene sequence is 50 to 60% similar with the banana, which is altogether a different species, and we share 80 to 90% of our gene. Our gene is similar to fruit fly, and almost 85% similar to mouse, though they are different species. Then this particular statement, like, the COVID-19 79% similar to SARS-CoV and all, when the entire health community is suspecting that COVID-19 is actually shares its genealogy with SARS and MERS. It is the symptoms of the infection is almost similar to SARS and MERS. Then these figures seems to be unreliable, and then I started. So after that, reading that blog, then why I read that blog, that is just by passion. I'm no way related to genomics. I just wanted to. I know a little bit of bioinformatics. This is just a passion. So immediately I went to NCBI, which I know I am very much familiar with this, because I have a series of videos on bioinformatics. Earlier days I used to download genomes from this, including a couple of other very renowned world databases for genomes. Then I found out the complete genome for coronavirus 2, This is Wuhan. I mean, it was, I think, this virus sample was obtained from one of the infected individuals from Wuhan, where this entire pandemic has blasted. And now I downloaded this, and you have to see my rest of the videos as how to obtain these gene sequences. I have a very detailed video in my YouTube channel, and I have that gene sequence in my here. This is the file. Actually, I mean, there are different ways to obtain these gene sequences. They all have kind of file formats, but in R, which I am going to use for this analysis, it is very easy to perform the analysis on any file of the type .fasta. And now when you go to this particular section here, you have a file, and then you have different types of files here. There are more than a dozen types of files. You have to download FASTA, and like that you need to get or obtain this gene sequence into your computer. You see here I have the three. I have COVID-19, MERS, this is not MRS, MERS and SARS. So now I'm going to perform analysis on these three gene sequences, and all of them are in .fasta format in my computer. Now coming to the application, now I have this application hosted in my GitHub repository. You just have to go to the github.com/kamakshaya, and here you will find a repo GAtRS. I call it as GAtRS, genetic analysis through R Shiny, something like this. This is the file which is very important for you. Anyway, this is a free and open source application. You just have to obtain this file by clicking here, and I have a separate video as how to obtain these files from GitHub repositories and open them using RStudio. Since this is a free application, I could not provide executable file or binary files, I mean entire library as an executable fashion. You have to download this file, raw file, and you can just click on this and see what is there inside. It's just a plain R program. As I said, this is a Shiny program. This is just a software program which is written by using RStudio R and RStudio kind of thing. RStudio is the editor, R is the programming language. And now you have to write, once you get that file in your computer, I closed my, let me open. So anyway, I have a very detailed video on all these aspects, like how to download them. And this is the this is the file GAT or something like that. Right click and then open with. If your computer has a file association, then this file will be identified with RStudio. Then you just have to press OK. I'm not doing this because it is already open in my computer. Now let me go to the RStudio. This is how you open. So when you right click it and open with RStudio, it's straight away. You will see that file in the left top corner of your RStudio. This is how RStudio looks like. The left top corner is a script file, and there are three other panes inside. Anyway, this is not a demonstration for. I'm not going to introduce you the R and RStudio. I'm just going to. You don't need to know anything about R and RStudio. That is what is the advantage of Shiny applications. Your application, you are going to open your application in your browser. Now once you have the file here at the right hand side, let me close this file so that you don't. I have only one file now, which I opened from my Windows Explorer. I mean, this is the window inside the Windows operating system. By the way, I'm using Windows operating system, which I don't like. I most of the time use. I'm a Linuxian open source software evangelist. I prefer my clients and students use open source software operating systems just like Linux. But the world, I created this in Windows because most of the people are not familiar with Linux. So I have to follow by the crowd. Now you'll find a small run button here, run app, something like this, at the right top corner of the left top pane. Now you just have to press run app, then this application will be opened inside the default browser of RStudio. This is just a RStudio browser. In case if you want to take this application to your default browser of the operating system, then you need to press on this button, open in the browser, something like this. Now I'm just going to continue in the same default browser of RStudio. Now let me introduce my application. This is very important menu, contact, where I have my details, because this is just a basic application I did not provide most of the methods in this application. In case if you are interested, if you are a dedicated full-time genome expert, you have to write to me or contact me by using these details. I will be able to provide consultation, or may be able to hand over advanced application which has almost all methods implemented through that. Now you have the home button. It is not very much important for you. Then I provided a menu called data, and here we'll be performing some similarities when comparing one genome with another genome. I mean one gene sequence with another gene sequence. That is why I provided two buttons. The first button, when you press, it will take you to the, you just have to upload the file. Now I am going to upload the COVID. This is a corona, this is a gene sequence of the coronavirus. You see here, this is the information of the, it is it's actually, what do you say, the short, very short piece of information of the entire genome. See here, omitted, you'll find the information like omitted to 28,881 entries. So this is the last row of the gene sequence, because R will not be able to show you. There are memory problems actually. There are other attributes here of the, they are not important for you. This is what is the information which we need to perform the analysis. These are all nucleotides, and if you are into genomics, I'm sure you must be knowing about what is DNA, RNA, genome, gene sequence, and nucleotide, and the base pairs. We also call them as base letters, or there are four important nucleotides: A, T, C, G, something like that. Adenine, guanine, cytosine, thymine, something like that. I'm not into genomics. I cannot explain you. This video, the objective of this video is to introduce the application, not the bioinformatics. If in case if you are interested, you can just write to me. We'll go to a special. Now I'm going to show you, this is how you upload the file into this application, and you also need to make the other file ready inside the application. Now I have both COVID-19, which means corona gene sequence, and also MERS gene sequence. Two gene sequences we have inside. Now let me thoroughly take you into the analysis. The very first menu is the GC content. This is very important metric in bioinformatics, especially when you are performing analysis on gene sequence of any biome. Then you should be knowing the importance of the GC content. GC content stands for the base pair of the nucleotides, guanine and cytosine. So this GC content, I mean there are a lot of it is highly significant metric in bioinformatics. One of the important observations in bioinformatics is that the stronger the metric, the stronger the virus genome, which means it is very difficult for the for us to manipulate the, I mean the stronger the virus, difficult it is to manipulate the virus. It is not the virus will not be amenable for medical interventions. It becomes a bit difficult to kill the virus, or maybe to get rid of the virus from the host physiology, something like that. Anyway, as I said, I'm not going to explain you the bioinformatics, but you can read it. But if you are watching this video, I'm sure you should be knowing a bit of genetics or bioinformatics. Here, this is actually the metric for coronavirus. The GC content in coronavirus is 38%. I don't think that is a very strong value, and the coronavirus is still, I mean the conclusion from this, I can conclude that the coronavirus is not as strong as we think. It's very the virus is very much amenable for diagnosis, and we can manipulate the gene sequence. I mean we can kill the virus, and we can also make fragments, kind of, I mean it is possible for us to dissect or make analysis on the gene. It's not that dangerous. If the value is close to 1, then we should conclude that the virus is very strong, robust, and it requires very novel methods to crack the the code of the gene, something like that. Okay, now this is there is a second option called counts. Now this will let you know the counts of the nucleotides. As you see, adenine, there are 8,932 um, adenine codons, and C cytosine, and guanine and thymine. Am I correct? Let me. Yeah, I think I'm correct. Adenine, cytosine, guanine, and thymine. There are 8,932 adenine, nucleotides, and 5,000, and so on and so forth. Okay, I'm not going to read. So this is the total configuration of the gene, and now these are the pairs of the nucleotides. See, we have. I calculated this. Application calculates this. Pairs with the help of kind of code, R code. We use our programming language to make this. Now I can see there are 2,859 pairs of AA, and there are 2,023 pairs of AC, something like that. Let us go to GC. This is very important. 1,168 GC and AT. Is there any AT kind of yeah, AT here, 2,307. So this GC should be minimum, otherwise the virus is not amenable for diagnosis, kind of thing. Now here the 3-letter pairs, 3-letter configuration, you have, I think the most important thing is ATG. The more you see here, ATG is there, 725, something like this. In 4-letter configuration, the most important thing is ATCG, that you should be able to find where is such it is. Anyway, this application is not going to do those searches. This will just give you the counts, and based on the counts we can come to conclusion and make some kind of inference as what is this. Now there is a GC plot. This plot by default, since this is a basic application, for every, this is a window plot for every 1000 segments, which means I'm dividing the entire gene sequence into 1000 parts, and each part is this is the GC content. The GC content calculated for every 1000. If we make 1000 the gene sequence, if we cut into 1000 parts, and we'll be calculating the GC content in each every part, and then we are plotting it. And you see the GC content in the very first it starts from here 1000. This is 1000, next 2000, 3000, 4000, 5000, and by 6000 this is going down. So the GC content is very weak at the 6000th part, and then it slowly rises and increases somewhere in between 10,000 to 15,000, and then again go down at 15,000. This is just gives a rough because the overall GC content may not be able to throw much information, so we should be able to make the gene sequence into the different parts, and then study the local value of the GC content, something like that. So it is roughly, I don't think there is any trend in this plot. It is just steady. There is a slight upward, that is why it is 38%, otherwise it will be downwards, kind of thing. Now there is a metric called rho. This talks about a kind of I may not be able to tell you the entire meaning of the rho, but here this is GC. This is a metric which talks about the importance of the commonality of GC content overall in the virus. You have a lot of documentation online, just like this. Here you see there is a value of 0.8651, something like this, which is very close to 1, and this means that the DNA here it is determined, they concluded that underrepresented. This is actually the representation of the GC content in overall gene sequence. I don't like to confuse you much, but you have a lot of information online, but here this is the GC content. I don't think this is under represented, and this is overrepresented. It seems like I mean, GC content across the gene sequence is not a rare thing, and it is thickly um, I mean constructed kind of thing. Now there are some similarity measures, and I would like to provide you the t-test. This is actually talks about the similarity between the two genes that we are studying here. MERS and COVID are the two gene sequences. So you can just make the similarity between the MERS and actually this test is it talks about the similarity of the means of these two uh, viruses, and now the t-value is negative and close to 0, and there is a p-value of the p-value is close to 1, so we cannot reject the null hypothesis that the mean value of gene configuration in COVID-19 or coronavirus is statistically, the difference between the mean values of these two viruses is not statistically significant, something like that. And this is a confidence interval, and there are means. You see the mean of corona and mean of MERS kind of thing, and there's a lot of statistics all around this, and this analysis is very much useful for academics and also in research to formulate the hypothesis and calculate the evidence in support or against of the hypothesis, something like that. Now suppose if you we can, it is also possible for us, in academics and research there is also kind of hypothesis that are they dependent or independent kind of thing, like I don't know how far it is meaningful in bioinformatics, but it is possible to have such kind of hypothesis. You see the p-value, if you have a hypothesis that there is a dependability between the corona and MERS kind of thing, so the p-value is also not in support of the alternative hypothesis, and we fail to reject the null hypothesis, which means they are independent. It doesn't matter, we can come up with an assumption, we can refute the assumption that the possibly a person who is infected with corona also can be infected with MERS kind of thing. So if we make such kind of assumption or hypothesis, and we can possibly reject such hypothesis that in fact both of these viruses are independent, there is no dependability between kind of. This is a mutual information, like suppose if you make the hypothesis that both of them depend on each other, like how much of suppose if both of them depend on each other, what is the mutual information dependability, how much of such dependability can be measured with the help of a metric kind of thing. I think I am confusing you a lot, and I don't want to get inside of here there are p-values, and we are just going to reject the null hypothesis that it is not possible for us to understand both of them together kind of thing. Now there is an association here, and this particular submenu must be able to compute all the. Now this is actually the aim of this. I started writing this software application now to know the similarity between the MERS and COVID versus SARS, and you see the similarity between these two viruses is 93.85, and this seems to be very reasonable for me because the similarity measure, the value 85 or something like I've shown you earlier. I don't know I have that or not. I think I don't have that. Somewhere I found that it is 85% similar and kind of thing. That is not true, I think, because the genetic distance between any two viruses must be very. Their virus is virus actually. Whether it is this virus or that virus, they are all viruses, and they must be very close. Even if this value 93.85% is ridiculous, it should be 99.99 kind of thing, because both of them belongs to same source kind of thing. Now this is Carl Pearson similarity index, like the similarity between this COVID, which means coronavirus, and MERS is 93.85%. Now this is the data which is used. Now when you look at the data distribution, you see There are 5,492 C's and 6,057 C's, and so they're all pretty close, right? So for such values, the making a postulation of 80%, 60% is not. It is a ridiculous value, kind of thing. Now let us change this. Instead of MERS, I'll go for SARS, and let's see the similarity value. Now if we go to the association, it goes to 99. So the coronavirus is 99.99% similar with SARS and only 93.87% similar with MERS. So it has, MERS is the distant cousin, and the SARS is the very near cousin to coronavirus, kind of thing. You see here the SARS, and at the same time, this particular metric, the measure, also depends on the source from where you have downloaded your information. These gene sequences are highly unreliable. No two gene sequences done by two different individuals are similar in fact, and they tend to have different configuration. Now it depends on from which source you have downloaded these data files. Your metrics are as best as your data, that's it, nothing else. Now let us go ahead and see some other. This is a pairwise similarities, alignment, and there are quite a few, and my system is a dummy machine, and it has only 2 GB RAM, and these methods are not going to work in your machine unless if your RAM size is greater than or equal to 16 GB. You can contact me in case if you really interested in this pairwise sequence alignment kind of analysis. I can provide you this particular matrix through consultation. Next one is multiple alignment. Again the same thing. Don't touch all these things, otherwise your application becomes down. You may break down your application, and then you have to close your computer. You can't go ahead and do the analysis. Now let me take the computational gene finding. I think there are a couple of other things, like here this is actually a standard what do you say, these are all I think this table belongs to a particular concept called match pattern. There are 3-letter configuration system in bioinformatics. Suppose if you look at ATG here, this is this particular combination of the nucleotides will leads to an amino acid called methionine, something like that. Will you able to find something called glutamine somewhere? Yeah, glutamine here. You see there is a combination of nucleotides, GAA, and they transform into an amino acid glutamine. So this particular system, this is called a standard table in genomics. This is actually a kind of we are going close to diagnosis kind of thing. Suppose if we study this matches, we'll be able to figure out how to crack down this virus. We may be able to gain more and more understanding on the virus, as how to attack and break down this gene sequence and collapse this particular virus with. You see here I have a particular ATG kind of thing, and this ATG, based on the the number of pairs of such match, this is just called a match pattern. Based on this particular pattern, we should be able to figure out whether this methionine formation happens in this gene sequence or not, kind of thing. So based on that particular observation, we must be able to figure out the method of diagnosis kind of thing, and treatment. Now you can go to the match pattern. Here you see ATGs. This is the start codon, and this is the end codon, something like that. So this is the place where the ATG starts, and this is the place where it ends, kind of thing. This is very particular and very yet very significant aspect in informatics, especially in genomics, to study the virus or any genome, kind of not only the virus kind of thing. So in this application, it is possible to make the match patterns, and you can just suppose if I use the TGA, then you will get altogether different. These are the strands where of the virus genome where your particular match is going to be present, or you will be able to kind of thing. And you can also change the other important match is TAA and kind of thing. These are all. And there is another feature called transition, and this is not available in this. See, not implemented. Upgrade to advanced application. You need to demand advanced application in order to perform transitions. Transitions are very very important. Converting the nucleotides into the amino acids. The actual work depends on this particular aspect of the analysis. Now let us go to the comparative genomics, and this is just t-test and all same kind of activity here I have shown you. And then you have hidden Markov analysis, and again this is not available in this application. You need to go for this is actually the some kind of probability distributions in statistical analysis, and here people will try to predict how likely a particular thing happen in a given genome or gene sequence, and what is the likeliness of picking up a particular nucleotide and how that is being followed by another nucleotide kind of thing. And here the people try to use hidden Markov models. There are a couple of methods here, and I'm not going to provide you through this application. You need to demand the advanced application. Okay, this is all about this application. I just wanted to create this application. I started doing this to verify the present observations in the domain about the similarities. I could convert that study into an application. This is just a basic application which might be useful for academics and researchers, and in case if you are interested in advanced application, just contact me using these details. Thanks for watching this video. Stay tuned to the new content, and don't if you like my video, please press like button, subscribe to my video channel. Thank you. Thanks again for watching.